Hello class, we are going to be working from pages 88 and 89 in your science notebooks today. We're going to be talking about mutations. So go ahead and update your table of contents for me. On page 88, you should have mutations notes. On page 89, you're going to write mutations output in your table of contents. Next thing you're going to do is go ahead and title the page mutations, and you're going to set up a simple chart with type of mutation on one side and example on the other, and you need enough for 3 types of mutations. Go ahead and set that up now. First type of mutation we're going to talk about is called substitution. The definition is pretty much as it sounds. So when a base pair or base is making a copy, one of the bases is accidentally swapped for another. So one base or nucleotide is swapped for another. This actually only is going to impact that one base pair. It's not going to affect the entire code. So if you have adenine, thymine, guanine, cytosine, adenine, and all of a sudden there's a substitution. So let's say adenine, thymine, and all of a sudden when it's making a copy of itself, it decides to substitute the thymine for a guanine. It's actually, because it's a substitution, it's just going to keep on going as if nothing happened. So it really doesn't impact the rest of the code. There isn't a trailing effect, and we know the DNA sequence goes on for a long time. Our cells are very complicated. Genetic code is actually interpreted in sets of 3 called amino acids. As you can see, as things get interpreted after this, there's no ripple effect. It's just this one sequence that is going to be impacted. So it might change something about the organism that it is creating, but it's not going to have a major impact and probably won't be a problem. In fact, when proteins come along to check this sequence, they're probably going to find that mistake and fix it easily. The next type of mutation is an insertion. And yeah, it comes from the word insert, so it means to add an extra. So an extra base is added to the sequence. This is going to have a major ripple effect, which I'm going to show you in just a moment. So if you have, let's say, thymine, cytosine, guanine, cytosine, adenine, thymine. Okay, and obviously that continues. Let's say it's making a copy of this sequence, and it starts off pretty good, and then it all of a sudden inserts an adenine. Now, because it's an insertion and not a substitution, this type is really bad, because the extra one is going to cascade the others. So you still have the remaining code, and this is going to go on and on and on and on and on. Except, ever since this is inserted, the rest are going to be interpreted incorrectly, excuse me. So you have amino acid interpretation. Notice every code after this is going to be different. The final type of mutation that we're going to talk about is deletion. And yes, it has to do with deleting. So one of the bases is deleted from the sequence. And you guessed it, it is going to have a major ripple effect. So let's say we have guanine, cytosine, adenine, adenine, thymine, 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 cytosine, guanine. Okay, so we know those are interpreted in groups of 3, and that sequence just kind of keeps on going. Okay, so these amino acid groups are important once again because if you have a deletion, so let's say it's the same until here, and all of a sudden this adenine is deleted, and so the rest just kind of slide over and fill its place. So this first sequence is okay, but everything after that will not be okay. There is a ripple effect. So this is where the problem started. This whole sequence after that is going to be gone or messed up. I like to compare these 3 to a sentence. So let's say we start with the sentence, the big man ran. Okay, it's a great sentence. If I wanted to make a copy of that, I would write the big man ran. Now let's say hypothetically while I'm copying it, all of a sudden the B is swapped out for a R. The rig man ran. It's pretty silly. But for the most part, I can see the words are going to be interpreted correctly, except for maybe the original word, although I would probably notice I made that mistake and fix it. So this one is your substitution. After this, there were no other problems. Everything else was fine. Then we have insertion. So let's say the R is inserted here. So it's going to have a ripple effect. So everything after that is still going to be interpreted in 3s. But the first word's fine. Everything after that is going to be changed. So starting with this, the rest is impacted. And lastly, again, sorry, that's insertion. For deletion, let's say I have the E deletes. Lastly, we have deletion. So if it started with the big man ran and the E's deleted, so the B fills in, and everything else is impacted. So after that deletion, that original deletion, everything else is going to be interpreted incorrectly. Again, these two have ripple effects, which is really bad when you're copying DNA because it's going to mess up the code entirely. Now let's do the output. So let's say you set up another table, except this time it's completely opposite of the first table. You've got example on one side and type of mutation on the other. So let's say the example is this sequence. Okay, turns into the sequence. So take a look. Which one do you think it is? Next, let's say we have the sequence turns into the sequence. Which type of mutation is that? And lastly, we have this sequence, and it turns into this one. So take a look, figure out which one it is, stay tuned for the answers. Down at the bottom you have one question to answer, which is which two types of mutations would have the greatest impact on the cell. You're going to write your answers there and stay tuned for the answers. So I am uploading a new answer key because the last one somehow the examples got switched around. So here we are. So if we look at this first one, the best thing to look for is where does the change begin? So if I'm going through, I notice the first change is here. So then I want to see, okay, how does that affect everything after it? So if things are interpreted in threes, then from this point on, there is a significant change in how things are interpreted. So we know that it was either a deletion or an insertion rather than a substitution. So if I look at this G and C, I can see that the C is carried over from the first one. So it looks like the G was just deleted and everything after that sort of slid over. So the answer for the first one is deletion. The second one, again, if we just start by looking for the first change, it actually starts a little sooner. Starts with this first group. So we can see that it is almost just like swapped out because everything else afterwards stays the same. So that's a substitution. And last but not least, if we look for the first change, it's right here. And if we look at how things are interpreted in sections of 3, we can see that it does alter, but the C is simply an extra base pair. The rest are sort of just pushed over. So that is an insertion. So which two types of mutations would have the greatest impact on the cell? That would be deletion and insertion. Doesn't matter the order that you put that. I'm going to put insertion, deletion. Either way, it's not substitution. Substitutions do not have a large impact on the cell. All right, that's it for today. Thank you for taking notes with me and doing your output. Don't forget to upload your video to Flipgrid.